So today, we are moving on to next topic. It's called Stereotaxic Surgeries and In Vivo Techniques. So today, before Chuseok and after Chuseok, we will get into this very practical aspects of neuroscience technology. So let me start with the Allen Brain Atlas. So hey, maybe. So let's see. Please show your face. So how many of you are like have seen the Allen Brain Atlas? Raise your hands. Okay. Okay. How many of you have not actually know Allen Brain Atlas? Have not heard? That's okay. You haven't heard. Okay. That's good. Majority of you haven't heard. So this is a good chance to talk about this. So Alan, you know, all of you know Microsoft. So the founder is Bill Gates, you know him. And the co-founder was Alan. So Alan donated money for the future of human, like, you know, the most important thing, that was the brain science. So he actually donated a big chunk of money. So that money has been establishing Allen Brain Institute. So that part is, it's in Seattle, and I went there when they didn't move on to a big building like now. There was a small like garage-like building at the time. I think it was 2012, so it's about 8 years ago when I visited there. So since then, they have recruited a lot of scientists. Like half of them are computer scientists, in fact, and you will see why. And in fact, there's a Korean project manager who actually made all this arrangement, especially the microscopic study of the mouse brain, to reveal all the microscopic detail of the neuronal connections. So that's what I want to emphasize, which is relevant here. So I had actually found this year's paper in Cell 2020 that they have published Allen Mouse Brain Common Coordinate Framework. So this, you see, it's in Cell. It is a long paper, and I brought it here. Okay, so for doing research, it's great to having an atlas so that scientists can communicate and having consensus of which brain region to explore. So because of that, they have made, so this is about 7 years ago in their paper that's showing this coronal section of brain. It's a three-dimensional information and showing gene expressions in 3D. Another one I want to show over here is, you know, this beautiful three-dimensional rendering shows the connections of the neurons, how do they connect each other. So what's very striking results after this project is, hey, the neurons we know in the brain, maybe they are in the regional effect. But what happened in mouse brain is even like single neuron can be, their extent to be throughout the whole brain. And think about the size of the mouse brain. Mouse brain is about your like thumbnail size, like about 10 millimeter across. And that is quite small. However, in microscopic point of view, of a resolution of a 1 micron, this is like 1,000 times of pixels. And so what it means is 1 million voxels in terms of 1 micron to describe, 10 millimeter is 10,000 and across. So this is a vastly big area for microscope to deal with. But what they've done is actually they've done that by using a new technology called serial to photon tomography. So basic idea is you watch having a brain and you take a 3D image, but you can't image it so deep. you remove that part out like a ham slice, and then the next one you take an image and you slice out, you take. So it's called slicing dicing project. In fact, this idea came from my PhD laboratory. So they found a company which this instrument was sold to Allen Brain Institute, like $1 million microscope, and they sell like 6 of these microscopes. So that's part of this story. And I encourage you to go to this atlasbrainmap.org. And this is a modern research. And when you're doing this research, there's a lot to explore. It's very fun. So that's your like homework. I mean, I won't check it, but you know, when you're time, just go there, take a look. I'll show part of it today. So what we want to study today is there are like a number. So especially for today, process of performing stereotaxic surgeries from applying anesthetic agent in Korean machi to monitoring recovery. Okay, what technique we will be covering is there are a number, but especially today, stereotaxic surgeries in animals. The concept of sterile fields and anesthesia, stereotaxic positioning of animals, accessing brain, implantation and recovery. More specific technical issues that implants that allow to the brain and how to measure neural activity, measure neurochemistry will be covered next time. So first, I guess you all now have experienced what is in vitro and what are in vivo experiments. So in neuroscience, we can also still bring that concept. So in vivo experiment is by itself. In vivo means a process is happening in living animal. That procedure take place in a whole living organism. So I put plus and minus as a pros and cons. So why we do in vivo experiment? Because it accurately reflects the physiology and behavior of a living animal, especially in neuroscience point of view. However, we must know and admit, for those who have done any in vivo experiment will realize how difficult it is. Okay, it's a very, very difficult, but that's worth it. That's why we do it, and expensive. And measuring or manipulating activity in intact organs and animals is relatively difficult. So that's why ultimately in vivo experiment is something like confirmative or like decisive. Just simply say, when you want to go to like high impact research output, that's what usually happening to demonstrate in vivo. Even if you have a very nice, great idea to prove in vitro, people would always ask, hey, so what? But there are reasons why we have to also do in vitro experiments too. So in vitro by itself meaning that you are performing experiments in a test tube, or this means in glass or Petri dish. And so it's outside of living organisms. So it sounds, you know, maybe not as great as in vivo, but remember in vivo it's very hard to perform. There are lots of variations and very difficult to control. So that's why we do also need in vitro controlled environment study to be able to draw a nice and reasonable conclusion relatively easily than in vivo. So I guess the first one I can tell. So in vitro experiment has a high degree of control over the extracellular environment. And also in vitro, you can perform accurate measurement of biochemical expression, because you can control where its biochemistry is. Usually you grab the cells, you grind the cell and measure the amount of neurochemicals or protein, RNA, DNA expression, and those will be easier with in vitro. I would say in vivo is not impossible, but in vivo is more complicated. But out of this, the results you get from the in vitro experiment may not reflect the complex microenvironment. So this is very important aspect. So let's say, for example, let's say you are studying, let's say brain tumor, and you can take out the tumor cells and grow on a petri dish, and you can measure some stuff, and or you are studying, let's say, trigeminal ganglion cells and their neuronal aspects on a petri dish. That's great. You can do very good tests, controlled environment, but in the end, the people will ask, hey, this environment is different from the inside the living organism. So, and that's the point. It's not always easy to draw conclusion, and you can't overestimate and overstate what you are finding in vivo, because that's only you can prove in vivo environment. So that's important when you have a great finding in vitro, which is great, and you somehow always think, people will always ask you, so what? So what's happening in vivo? So are you sure that what you're finding is important in real living animal? You want to prove that too. So when you have a great finding in vitro, you will need further verification in vivo experiment. So that's like, not share about the prelude. So now we talk about stereotaxic surgery. Why accessing the brain? Okay, so maybe I'll start with the Jibam. Okay, let me show you one picture of the stereotaxic device. You can see, wow, this is a little gross figure, but you see there's a mouse, the brain or scap is exposed. You have to fix the mouse brain with these strange bars, and even these pins are supposed to like pointing it. So we call stereotaxic surgery a stereotaxic surgical instrument looks like this. Then what is stereotaxic procedure, which we want to sometimes a substance to inject into a discrete brain region, and this device can be also implanted inside the brain onto the skull. So just the literally what it says, stereo means 3D and tasic means arrangement. So maybe we can have a Jibam. You can guess what to do activity and neurochemistry of cells in vivo. We want to use stereotaxic surgery. Can you guess what this is? We want to probe the activity and neurochemistry. Okay, yeah, that's not easy. So that's to measure activity. So we do experiments precisely, and we want to use stereotaxic surgery. And the second, this is something like we observe and measure. We want to kind of look at and read it. So the other aspect of this study is also we actually get actively intervene neural activity so that to determine the effects on behavior. For example, we could send out electrical current, okay, to stimulate, or optogenetic simulation, which requires us to send the light to stimulate. Jibam, can you guess what this menu? Exactly. So look at these two different kinds. We could measure, we could manipulate, and in a distinct brain region. So that's why we use stereotaxic surgery. So stereotaxic surgery is by itself, unfortunately, the brain and spinal cord is called central nervous system, which is protected by skulls and like bones. So it's inherently invasive procedure using precisely target specific regions of the brain. So let me show you one example. So in the after Chuseok, or throughout this course, hands-on, we will provide you have a chance to actually perform stereotaxic surgeries. But after Chuseok, there's a hands-on stereotaxic experiment for except those who are maybe not in GIST. If any of you have a concern about watching this or touching animal, and due to the COVID, if you don't want to take part in the hands-on, please message or email me so that make sure we don't want to force you, but there will be a total class section for demonstration. And so like, I hope everyone would agree, but if any of you are uncomfortable or not able, please inform me and TA, okay? So we will respect your any of your concerns. All right, so you see these ones. This, in fact, is a rat. How do I know? Because of this nose part is actually bigger. So you see the scar cap is being exposed, and to make it being exposed, you can even use some of this tools to keep it open. And you see these are drills actually making a hole onto the scar. So stereotaxic surgery, placing animal brain, we have a 3D coordinated system to place neurobiological probes such as neuropeptide or neurotransmitter, or even virus and reagents in discrete brain regions. So here comes a little more detailed view of the stereotaxic device. I would say you have to know all of this, okay? So you see, look at that. There's a nose bar here, and there's a ear bar, left and right. And also underneath the nose bar, there's a hole in it. So this rodent has a very big frontal tooth, okay? So you actually use that one to fix. So that's we call bite bar. And for stereotaxic, you need to have a three-dimensional control and targeting. So what do you need is X, Y, Z translator, and even that's a precise translator. So I will say to you later on, so we have some stereotaxic arm and metal grip for needle, so that you can inject something and point some in a right place. And that arm is on top of this dorsal ventral reading, medial lateral reading. I would say this is wrong, so you can figure it out why, and then anterior posterior reading. So just to look at the shape of this, you see this is frontal back, it's moving, and this one ups and downs. In fact, ups and downs in this is in fact dorsal ventral. Okay, and this is from lateral part, and that's medial lateral reading. So that arrow has to be here. This arrow is kind of okay, but this one, right? And hopefully it's clear to you. Oh, so the last one, hey, so 3 points are necessary to maintain rodent head. So 2-year bar. The problem of 2-year bar is you can move your head ups and downs, rotating. Because of that, you need to have this frontal tooth. It's called incisor, okay? So incisor to make 1 to 3 points are necessary to fix in 3 dimensions. So that's usually we use upper incisor for the rodents. Okay, now I have to say there are a lot of terminologies, so that's why I ask you to preview. It's important, especially when you're not very familiar with this. Let's see, Min-sung. So I guess you can answer this. There are 3 planes to section and orient the brain. So this green section, can you say what this is? Let me, yeah.
Corona.
Okay, great, right. So this section is called coronal section. And what about this frontal and lateral?
This blue one?
Yes, blue.
Sagittal.
Okay, great. Yes. Note that there's a two double T, okay, when you write it down. And the rest part is horizontal. So you can see it's horizontal, okay? But in CT and MR field, people use sometimes different terminals. So you better to know that. So horizontal can be also known as a, Winsome, can you also guess?
Transverse.
Okay, great. And the last one?
Axial.
Yeah, this is a bit hard, but yes, that's axial. Okay, so you have to know, like, when we are cutting to show the section, we specifically use coronal, sagittal, or transverse or horizontal sections. And also, look at in the human brain, on you can see there are like different terminals, which I will discuss it next slide. Okay. Okay, so that's in stereotaxic frame. So this is a human brain, and also you can see this is a cerebellum, or like small brain over backside, and there's a spinal cord. So then the spinal cord can have this cut, coronal kind of coronal cut, and you have to see here. This is imagine there's a human there. So this is the backside. So back in Latin is called dorsal, back. And then or belly side is called ventral. So that's how we define dorsal ventral. And that goes together into the brain while there's no belly, but in brain, you see the upper part is called dorsal and the lower part is ventral. Of course, there are other ways to say superior and inferior. Okay. And then frontal and backside is obviously, we already know this is called anterior and posterior. But there are other, especially when we are dealing with animals and rodents, there are other terminologies because the anterior part is originated from beak, okay? That beak is called a rostral, okay? And then in the other part is tail. So tail is called actually caudal. So, you know, this is the terminology you must know so that you can read literature and communicate. And dorsal ventral, I already said. And the last part I want to explain is actually most of these things are symmetric. So we want to define from the center to the outwards. So towards center is called medial. Okay, towards outwards is called lateral. So like here, medial, lateral, you can see, and even in this coronal section, you can find which part is medial and which is lateral. So medial and lateral. So hopefully this makes you sense. Of course, there are all Latin, origin, which is kind of you have to know, and maybe this can help to understand and memorize. I'm not talking about when you use often, you will memorize it automatically, but better to know now so that it's easier later on. So Brain Atlas providing useful 3D guide for targeting discrete brain regions. In fact, in many neuroscience labs, targeting brain is a difficult job because it requires a skill to develop, okay? And Stereotaxic Atlas, I want to show, and anatomical landmarks is an example is, so this is the book, when you have a book of Atlas, now the book is all in PDF though. So this is the Atlas, and this is the skull of the, let's say this mouse or rat rodents. And what you want to see here is actually that the Bregma minus 5.40. So this is a special section, a coronal section. And how do we locate? Requires a good landmark. So if you look at the brain skull, and you'll realize there are landmarks. So this is called bregma, and this is called lambda. Okay. So, and you see these skulls when they form a bone in the skull. So there's a seams in it. Okay. So that we need to label them and know that. So we can identify the following. So you can see the frontal side of this suture. There are frontal, actually the frontal suture, and you see this like coronal section. It's called coronal suture, and these lines goes along the sagittal. So this is called sagittal suture, and you know the back in the brain on the back side there's an occipital lobe. Okay, so that means in fact it means against the head. Okay, so that part is called occipital suture, or the shape in geometry looks like rhomboid, so that's why it's also called rhomboid suture. All right, so then maybe I can ask you for further. So the most important landmarks are in fact bregma and lambda. Why? When you open up a living brain skull, and you will see like, oh, where to start? So there's this point and this point. So because A, B, C, D, B is front, then L. So I usually remember that way. So frontal part is bregma and backwards is lambda. Okay, so that anatomical landmark can be visible. And bregma is intersection between, you can see here, let's see, 최유진. Okay, so bregma is intersection between sagittal and exactly coronal sutures of the skull. And however, when you actually look at under the scope, you realize it's not that clean. So even that's not as, it's simple to say conceptually. If you look at it microscopically, you realize it's not that easy and simple. Okay. And what about lambda? The intersection between the lines of best fit through this rhomboid suture, which is here, and Yunhe, 최유진, again. Yes, exactly, sagittal. And the problem here is, in fact, you look at here, it goes this rhomboid suture is in fact not straight line, and it goes this. Okay, and this is sagittal. So you realize there are problems and issues. So that's why we use the lines of best fit for defining this lambda. Okay, so due to individual variation, at the same age animal will have a pretty consistent, but you know, individuals has variation. So correct targeting needs to be confirmed. So let's say you are doing your thesis work, your study, you target, you believe you did it the right way. Of course we believe, but like later on, how can you persuade others, reviewers, and your professors, your thesis committee member? How will you prove that you did it the right targeting? 최유진, can you guess? Confirmed by examining what sections of the brain after experiment has ended? Okay, it's not that difficult, but okay, that's histological section. Yeah, it's difficult. So what it means is, hey, you did it in vivo living animal, you did a good job and targeting and did blah, 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 manipulating and, you know, injecting substance. But later on, like people will ask you, how do you confirm? So you take out the brain and make a section and actually reveal and show, prove that you did the right targeting. Okay, good job. So I just want to broad how we use Atlas when you are actually doing targeting in reality. So you have this Atlas book, and then this is one example I brought so that, hey, you know where you are targeting is this location. Then you have to first see from the bregma 0.72 millimeter. What that means is in the bregma, okay, bregma 0.72 millimeters away, and this is coronal section, you see. So from 0 to frontal part, about 0.72 millimeter, and then you cut. So that's this location. And then you need to have a 3 scales for targeting. So this ML, which means medial lateral. So from the center to the right side, plus or left side, 2.9 millimeter. And then you have to define the depth. So you have to touch at the top of this, okay? And then you go down, okay, actually this is 0, and you go down minus 4.4 millimeter, then you can properly target. So this is typically how you use this atlas in targeting using stereotaxic frame. And actually, yesterday I brought this one. Sorry, it's not included in your, but this is only reference. So exactly, so this is a traditional book you have, and you use this targeting. Now, the one in Atlas, in the brain Atlas, you go there and you play with it. So I was trying to find the most similar to this from the Alan brain atlas, so which is in this paper, right? In this paper. So this paper is about using like 1,675 mouse born at day like 59 days, and use many mouse to make an atlas so that people can design and experiment and confirm their findings in exact brain atlas. So look at this. I can only show this, but if you click, the proper names and locations are all in digital format. So this is great. So that's why I want you to play with it. Okay. So how to position a rodents on a stereotaxic instrument. So important thing is flat skull position is important. So when you have this skull into stereotaxic and you realize it's hard to make, how to make ensure the skull is flat. So you have two landmark called bregma and lambda, and when you have to target it, then you have to make the bregma lambda in exact position. How you do it? You use stereotaxic to pinpoint bregma, and then going back to touch lambda. If it's high, you rotate back, and then you make this 0 and come back bregma, and there will be off. Then you go back and forth, okay? Iteration to make it flat. So to ensure proper orientation, otherwise you will have an error when you go deeper and deeper. So placing the head such that top surface of skull is flat from bregma to lambda. So that's in rostral and caudal direction. And how would you access the brain? So after your head is flat skull position, and the discrete brain region can be targeted. But to access the brain, you need to cut the skull off with a dental drill to create small hole or a small window in the skull to expose brain regions of interest. And that part, let me see, Zhu Jihun?
Yeah.
Can you guess what this is? Cranial?
Radio anatomy.
Great, yes, it's thumb. Thumb means cut, okay? So it's called, cranial means a, skull, okay? So you have to cut the skull. And something you have to notice, removal of dura and thin meningeal layer of tissue surrounding the brain. So, so the skull and brain, there's a thick, it's like a rubber latex, like one for human, that's dura, okay? So that's there, and then in this, it's called a meningeal layer. And sometimes we leave dura intact, sometimes we remove the dura, depending on the purpose of the experiment. Okay, so for this, I want to explain you a little bit of the multiple layers from the skull to the brain parenchyma. parenchyma means the business end of this organ. Okay, so I want to show the outer lining of the brain. There are a new terminologies you must know here. So there's a skin and there's a bone. And as I say, there's a three major linings between the skull and brain. That's dura mater, and arachnoid mater, and pia mater, and the brain. So first, pia mater is the outer surface of the brain parenchyma. Okay, so this is a real outer like coating or surface. And beneath the bone, there's a relatively thick dura mater is there. There are blood vessels inside. And here it's a very simple say arachnoid, but arachnoid is a kind of space. People use subarachnoid space, which is not clearly shown here. I will show you that. And one thing I want to emphasize is when you do craniotomy and to make an implant on the skull, you have to make sure to really clean the bone, the skull surface, which we remove periosteum. So this is a place where progenitors of a bone. So when there's damage in the bone, there are stem cells here and even blood vessels here. But this has a lot of fibrous collagen layer, which is not good when you implant because it'll kind of detach later. So you have to remove periosteum. Okay, and the unfortunate thing is periosteum is where the sensory nerve endings are there. So remember, bone, when there's a like damage in the bone, it's very painful. In Korean, 뼈를 깎는 고통. You remember that? It's very painful. That's actually due to this periosteum, all right? Okay, so let me go a little bit deeper into this. And I just want to show this periosteum. It's on top of the bone. And then beneath the bone, there are meninges. So we call meninges are like these three layers, dura mater, and arachnoid mater, and pia mater right on top of the brain. And you see arachnoid, a subarachnoid space is, as people call this space between arachnoid mater and pia mater. Okay, so how does actually it look like? So if you have a 2-photon microscope, you actually can see what's in microscopic detail. So let me show you for this. So this is a cartoon, and this is real image in a vertical section. You see, when you use a microscope, you can actually see collagen fiber, which is shown in blue here, okay, by so-called second harmonic generation. And then you can see the thickness of the bones left on top, so skull bone. And there's a space here that is, and it's a black, so that's a subarachnoid space. And what about the, there are many blood vessels, so that's shown in red. The real brain, you see the brain outer layer is called pia mater, and especially right underneath pia mater, there are glial limitants, which is lots of astrocytes are actually covering there. So that's why you can see here glia limitants. Okay, so that is the power of microscope, actually looking at the brain and astrocytes, and you can see glial cell type called microglia, which is a kind of immune cells inside the brain, and macrophages, and also stroma cells and fibers. Okay, so you can see with this microscope to see how this brain is composed of. And when you do craniotomy, by using this, we can exactly measure, let's say if you have a thin skull, we can exactly measure the thickness of the skull you make, the quality of the surgery you've made. Okay, so now go back to why we are using rodents for these surgeries. And yeah, so because it's similar, why we are using rodents? because the ultimate goal is we are interested in human, because the nervous system is similar, okay, rather than invertebrates. And it allows larger sizes than larger animals such as cats, primates. Why not non-human primates rather than these rodents? Because when we do science, statistical power of experimental results are very important. With just a few animals, it is not easy to draw a conclusion. Okay, so that's why, let's say Zui, can you guess what this is?
Yeah. I don't know this.
Yeah, larger sample sizes. Okay, you remember the Q&A session, number of n. So mouse breathes quickly. So we can quickly have lots of numbers. So you saw the previous Yunhe's presentation, the number of animals are even like dozens. It's impossible with the cats or primates. So for the remaining time, I'm gonna talk about this part of this creating sterile environment and anesthesia and positioning rodent on a stereotaxic instrument and assessing, penetrating the brain and finishing surgery. So how many of you have not done brain surgery? Raise your hands. Okay, let me see. Who have not done surgery, brain? Okay, great. That's fine. Yeah, so those will have a chance to do brain surgery yourself so that you will become a neurosurgeon of rodent, okay, after this course. Hopefully that will be an exciting experience for you. So, but like a lot of these animal surgeries, what I've observed is in neuroscience, people may not care too much about the environment. So for example, when you create a craniotomy and make a window, it lasts, you know, if you do it well sterile, it can last until the death of the mouse, which is about like 2 years older, several months, several weeks. But if you do not make sure this, you know, only a few 1-2 weeks after, you will find something growing underneath the scar, underneath the window, and you may not be able to have a long-term experiment. So all these knowledges should come from a human surgery, okay? So that's why the sterilization and sterile environment is important. So I have a couple of pictures from my laboratory that we do have this autoclave. So this is a strong heated container for high pressure and temperature. So the important part is really ensure all the dealing with killing and removing all microorganisms. You need 2 aspects: high temperature, high pressure. That makes sure. So autoclave is the thing you put your instrument, you can be sure that this is great. You can go to touch inside a living organism. Okay. However, you cannot do it in the middle of experiment, only before the experiment. So in the middle of experiment, you can use this kind of tool, which is heated up to maybe 250 degrees Celsius. You can have this one tool between your surgery. So that is called a glass. Let me show you that beads. Oh, okay. Bead sterilizer or heat sterilizer, okay? So that's a quick and easy way to sterilize your surgical tools and instruments. And think about when you are getting surgery at some point or in your life. So this is how the surgery has been done. And you know, this is surgery room, and you know, you can ensure bacteria, you know, bacteria are around. How to ensure your room is all sterile? You can't, but at least what you make sure is the surgeon having the costume, the surgeon has to be sterile, and the place over here, when the patients or the animals are there, you see these places and these tools and nearby. that green area we call sterile field, you have to make sure that is not contaminated. For those who have a chance to perform in vitro cell culture experiments will know clearly a little better about contamination, how easy you can get contamination. So this is a way you make sure this part, at least not other or bottom floor, at least these parts are where you can safely put your instrument there and then take back and then do the surgery and put it there. So that's you have to make sure. It's called sterile. So what do we mean by sterile? Sterile is not only like means someone cannot produce offspring. And this sterile is a little different. This means clean, but not just clean, but also called, let's say this is a little difficult. Maybe Una?
Aseptic.
Okay, great, yeah. Aseptic. This means is free from microorganisms that can lead to infection. So that's, you could sometimes use your tool just in alcohol and then you believe it's clean and sterile enough, but it may not be true because let's say your instrument having pores or fungus that may still there with an alcohol. There are microorganisms that love maybe living in alcohol. So you may not trust the alcohol fully sterile, okay? Aseptic means there's no microorganism. So remember, a lot of people die in the hospital due to sepsis. Sepsis means microorganisms, especially bacteria, growth inside the blood. Okay, septic shock, or in Korean, it's called 폐혈증. It's a big part of the end stage of patient die. So that's what you don't want, that any of microorganisms alive in your surgical field. So any invasive, you cut and reveal the inside of the body, surgical instruments and probes, as well as the surface has to be sterile. That's the point when you have the surgery. And two ways to sterilize implant and tools is, as I already said, it's autoclave, which is applies to increase pressure and heat, okay? To kill any microorganism on tools and reagent. But there are some cases you may not be able to autoclave, such as, let's say you bring your microscope into the sterile field, then you have to do somehow very strong chemicals to really ensure things are sterile. And heat sterilize, I think I already told you, but it's between surgeries, you could use it for temporary sterilization. And as I said, here is a dedicated surface as a resting place for tools when not in use. It can be clothes, it can be paper and gauze, and that's sterile field. Okay, so now I want to go to further anesthetic agents for animal experiments. Okay, first, ketamine. Ketamine is the anesthetic, and this look like this. It's a controlled substance, so you can like keep it in your own pocket. Okay, it's controlled. And xylazine. And a commercial brand of xylazine from Bayer is called Rompun. So it's also known as a Rompun, but Rompun is a brand name. It's xylazine. And these are liquid anesthetic, which means you inject into the rodent. However, another aspect is we do use gas for anesthesia. So this is gas anesthesia. It has a little bit more. You can see this this part where you use gas anesthetic. You have to make sure it's always not empty, not to wake up the animal in the middle of experiments, which would be very painful. And then you have this nose piece so that it keeps delivering this anesthetic in a humid and compressed air. And then before you put this one here, you have to kind of for a few minutes of the animal to be like really unconscious so that you can put it here. So that's called inducing box over here, and there are heat. So anesthesia means insensitivity to pain, especially artificially introduced by administering gases or the injection of drugs before your surgical operation. Remember, there's a pain involved in surgery, okay? So injectables are usually for short surgeries or experiments. And as I said here, as a, let's say, it's called ketamine. Ketamine is an anesthetic. So more details are here. But one thing I have to say is ketamine is not used for human much, but I heard people that sometimes they use ketamine for pediatric patients. And ketamine is known to cause nightmare. Okay. I haven't got it myself, so I do not know, but that's what's known. And ketamine is not usually used alone, but usually combination with xylazine, okay? And you see xylazine is sedative and muscle relaxant and analgesic. And it's agonist for beta to adrenergic receptors. So these two we use as a cocktail. So you will see in many papers, K and X, ketamine and xylazine cocktail. And you have to remember this combination you inject, usually it lasts about like 30-40 minutes, and you have to check the animal status, because if your surgery or procedure or imaging lasts longer, you need to have a boost in the right time. Otherwise, once it gets awakened, it's hard to get back again pretty soon. So, and I give my previous dose, which is 100 milligram of ketamine and 10 milligram of xylazine per kilogram body weight. Okay, so whenever you perform your experiment, make sure write down your dose, because this is very, very important to know, and people have a little variation from field to field and group to group. All right, the second is gas, which is more longer surgeries or experiments. Yet the good thing about injectable is easy to inject, so the animal knocked down and then you do experiment. The downside is when waking up, it takes a longer time, okay? The recovery time is longer. But gas, the good thing is, as far as you put it there, the animal is under anesthesia, and when you take it out, very soon the animal will recover, okay? And one of the most popular ones is called isoflurane, okay? And that's inhalable ether that likely works by disrupting synaptic transmission. You see, likely, what it means is we don't know the mechanism very clearly. Okay, so this is kind of a mystery. There aren't many anesthetics found in human history. And as I showed you, it needs an induction chamber, a small ventilated chamber, and you place nose mask for steady supply of oxygen. So which is, you need to make sure the air flow rate, if things are not right, you can awake the animal. And so that's very important. And then how to check that the animal is unconscious or not. In general, under anesthesia, the animal body cannot regulate their temperature very well. So you need to prevent, let's see, Kohina, can you guess what this is?
Hypothermia?
Exactly, yeah. 저체온증. So you want to make sure the animal to keep their temperature. So by putting a heating pad underneath, yeah. So animal can die under anesthesia. So you have to make sure. And also you can do tail pinch, 꼬집는 거. You can pinch the legs to find withdrawal reflex is gone. Then you kind of know. There are various ways. Okay. Okay, so as I said here, like ether, I want to tell you a little bit of a history of anesthesia, because ether is the first anesthetic which was kind of people figured out. Before knowing this, unfortunately, when we do human surgery, we just cut the human actually without anesthesia, so it's painful. You know what? Even people consider female feels less pain, so they actually use female as an experimenter. Like this is like history. So when we found out ether as an anesthetic, it was like kind of magic. So it was first used in the US. This is the place called Mass General Hospital. When I did my postdoc, it's Harvard Medical School in the oldest hospital. And here comes the seminar. But in the other end, this is tour place, because ether was first demonstrated here to be used as a surgery. So you see there's the mummies from Egypt, and there's a picture. This is the picture in the past. 1846 was first demonstrated here to be used for human surgery, and afterwards even human surgery, hey, without pain. This is great and amazing thing, and it's a history. So I just want to show you this picture. And now penetrating the brain, once brain is exposed, or you use micromanipulator to deliver a bioactive substance or neurobiological probe into the brain, either pharmacological, or you can inject the virus, and by using cannula or optical window. You can have a permanent implant, and you do need to use like dental acryl or bone cement. There are various things, expensive to cheap, good or not as good. So it requires a more experience. And when you finish the surgery, it's important to have to seal the incision, and you have to clean the area, help the animal recover. How to seal? You have to use sterile suture, or even sometimes tissue glue is there. And that part you prevent in further infection by using iodine or betadine solution on top of this sealed skin. And heat, you want to make sure, especially injectable, the animal take like even up to an hour, several hours, you want to keep the animal warm, because they may not recover soon, or even the blood loss and heart rate is low. So you can put recovery cage on a heating pad, or using sometimes warm infrared light. Aftercare, you need to use sometimes analgesics or antibiotics with proper monitoring. And you know, the animal you care is the most important, precious experimental animal. You keep it until the death of the animal. So you want to keep, like, it's supposed to watch them every day, in fact. When you especially did a surgery, pain or discomfort, you see when they are not moving, when they're hunched position, that's a sign that they're very, very sick, okay? Lack of eating and drinking. So you want to make sure that. Interesting, the animal doesn't seem to show their pain easily to human. Maybe that's their nature, because they don't want to show their distress to their predators, maybe. So somehow when they actually show signal or body weight decrease by 10%, 20%, you have to know, okay? So today I discussed brain atlas, stereotaxic frame, Bregma lambda, and surgeries. And next we will talk more about this measurement and manipulation. So thank you for your attention. And